My name is Leah Phillips. I am a student researcher within BioSIF, which stands for the Biophysics and Optical Sciences Facility at Appalachian State University. I was awarded the NC Space Grant Graduate Research Fellowship, and today I'm going to talk to you about what laser tweezer Raman spectroscopy is and my role in automating our system. So first, I'm going to begin by giving a brief overview about what Raman spectroscopy is, what laser tweezers are, and then I'll conclude with what the focus of my research is. So Raman spectroscopy is an effective diagnostic technique where we can shine a laser light onto a sample, and then we can examine the light that's scattered off. The largest portion of scattered light is what we call Rayleigh scattered light, and this means that it has the same energy as the incident laser. Out of all the scattered light, there's a fractional amount which has its energy shifted, and this is called Raman scattering, and this is what we're interested in. The change in energy is due to the sample's molecules' specific vibrational modes, and this provides a compositional fingerprint that's unique to other materials. So we use this in our lab for the identification of materials and samples. The second method is laser tweezers. Laser tweezers are a practice that utilize laser light to trap and direct microscopic objects. So for an incident laser on an object, there are two possible results. We have scattering and then we have refracting. The force from scattering models this transfer of momentum between the incident photons and the trapped object. So for simplicity, when we think about spherical objects, we can imagine this scattering as being a reflection. So we have the sum of the forces of the individual photons will add up and it will just propagate in the same direction as the incident light. This pushes the trapped object upstream of the laser, almost like a fire hose. Conversely, we have the refraction force, which we call the gradient force. And this is more of the tweezing action that we think of when we think laser tweezers. As light refracts through the object, the incident photons' momentums experience this shift in direction. And to conserve the momentum of the whole system, the bead or the spherical object that we're trapping must shift its momentum to be equal and opposite. And this reaction ultimately leads to the bead to be centered onto the laser, and this is what we call a stable trap. So here I have a video of our laser tweezers in action, and this is all custom built by students in our labs. So these beads are roughly 2 microns, and we can trap a singular bead and then guide it through our sample. And you can see that the stationary background objects or the background objects stay stationary, and this is all done with a laser light, which is really cool. So the focus of my research combines these two methods, and this is what we call laser tweezer Raman spectroscopy, or we'll call it LTRS for short. It's helpful in a variety of areas. So as I mentioned previously, the scattering force of light acts as this fire hose, and it pushes the trapped object downstream of the focal point, and this is not ideal. The focal point is where we get the strongest return signals for Raman spectroscopy, and this is where we want the object to be preferably. Raman signals are already weak because it's only a fraction of what the scattered light is, and it's often overwhelmed by outside noise. So this is where we bring in the trapped laser, the trap laser. So what we do is we trap the object that we want to study, and then we can move it back into this focal plane where the signals are maximized. And this increases the return signals and helps diminish outside noise, and it's overall a very helpful technique to utilize. The problem comes in when we keep adding more components to our system because we have several beams and we add mirrors, we add lenses, and overall aligning is a very difficult process that has to be done carefully. So this is why automation of our apparatus is so important. I have been primarily working to automate the laser tweezers. I'm doing this by attaching motors to the lens that is responsible for the alignment of the trap laser. So what this does is it aligns the trap laser on the object, and then we can move the object back into the focal beam of the Raman beam. And it'll make these micro level adjustments that me as a person, I can't make, and overall is much more precise. We're doing this through custom LabVIEW software using a simplex maximization method, and it puts it into the optimal position to collect Raman spectra. An automated LTRS system has broad applicability to the research and development of uncrewed space missions. It's overall a safer option that can greatly reduce human contamination and overall allow for analysis to be done without the transporting of the sample back to Earth. And overall, it's a real asset to future space exploration. I would like to thank you all for listening to me today, and thank you to NC Space Grant for giving me the opportunity to speak. Thank you.